Hi everyone, welcome to Baggy Infotech Media. I am yours KNS. This talk is to explain genetic engineering or so-called recombinant DNA technology. The first part of this talk mainly focuses on the tools of genetic engineering. Sometimes a good idea comes to you when you are not actually looking for it. The below is the popular saying by Dr. Carrie B. Mullis, the inventor of polymerase chain reaction. Later on, during 1993, he bagged the Nobel Prize for his invention. Here we mainly see about DNA and the central dogma of biology. What may be the base for the rDNA technology? That is nothing but nucleic acid and DNA. Probably you may know about the structure of DNA. DNA is made up of two linked strands that wind around each other to resemble a twisted ladder, a shape known as a double helix. Each strand has a backbone made of alternating sugar, that means the deoxyribose and phosphate groups. Attached to each sugar is one of four bases: adenine, cytosine, guanine, or thymine. Then, what is the central dogma of biology? From existing DNA to make new DNA, that is called as DNA replication. From DNA to make new RNA, that is called as transcription. From RNA to make new proteins, that is called as translation. What are the possible transfers in central dogma of molecular biology? This was well postulated by Crick during 1970. Number one, the arrows show all the possible simple transfers between the three families of polymers. They represent the directional flow of detailed sequence information. Number two, the arrows show the situation as it seemed in 1958. Solid arrows represent probable transfers and dotted arrows possible transfers. The absent arrows compared to 1 represent the impossible transfers postulated by the central dogma. They are the three possible arrows starting from protein. Number 3, a tentative classification for the present day. Solid arrows show general transfers, dotted arrows show special transfers. Again, the absent arrows are the undirected transfers specified by the central dogma. What do you mean by a gene? The basic unit of heredity passed from parent to child. Genes are made up of DNA sequences and are arranged one after another at specific locations on chromosomes in the nucleus of cells. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins. Most eukaryotic genes contain segments of coding sequences called as exons, interrupted by non-coding sequences named as introns. Both exons and introns are transcribed to yield a long primary RNA transcript. The introns are then removed by splicing to form the mature mRNA. The structure and chemical composition of the DNA in both the eukaryotic and prokaryotic cells are different. The prokaryotic cells have no nucleus, no organelles, and a very small amount of DNA. On the other hand, eukaryotic cells have nuclei and cell organelles, and the amount of DNA present is large. Now you know about DNA, central dogma of molecular biology, and what is meant by genes. Then one can ask the question of what is meant by genetic engineering. The answer is simple. Recombinant DNA technology is also known as genetic engineering. This means altering the genes in a living organism to produce a genetically modified organism, GMO, with a new genotype. Various kinds of genetic modification are possible, inserting a foreign gene from one species into another, forming a transgenic organism, altering an existing gene so that its product is changed, or changing gene expression so that it is translated more often or not at all. Do you think rDNA technology is chimeras? You know what is meant by chimeras? Impossible to achieve. But the nucleic acid manipulations can be achieved by propagation, ligation, digestion, addition of phosphate or methyl groups. And these modifications can be catalyzed by enzymes like polymerases, ligases, nucleases, phosphatases, and methylases. What are the tools used in genetic engineering? On top of it, the primary tool used is restriction enzymes. I just refer this as a molecular scissors because they mainly used to cut the DNA sequences. Who discovered this first? Stuart Lin and Werner Orber. The host mainly uses restriction enzymes, especially the bacteria, for its self-defense. The first restriction enzyme, it was discovered and named as endonuclease or later renamed as HIN2, and they mainly used to degrade the bacteriophage T7 DNA. So later the Nobel Prize in Physiology or Medicine for 1978 was jointly given to Werner Orber, Don Nathans and Hamilton Smith for the discovery of restriction enzymes and their applications in genetic engineering. Then you may ask the next question, how do restriction enzymes work? Like all enzymes, a restriction enzyme works by shape-to-shape matching. When it comes into a contact with a DNA sequence with a shape that matches a part of the enzyme called the recognition site, it wraps around the DNA and causes a break in both strands of the DNA molecule. The best example is the EcoR1. The cutting site you can able to very well refer from here. As I said earlier, restriction enzymes is from bacterial origin. The enzymes that cleave foreign DNA are named after the organism from which they were derived. The few examples are EcoR1 from Escherichia coli, BamHI from Bacillus ameloleukophaciens. They mainly protect bacteria from bacteriophage infection by restricting the viral replication. There are 4 different types of restriction enzymes according to their mechanism of action, subunit composition, and cofactor requirement. New England Biolabs produces and supplies recombinant and native enzymes, reagents for life science research. They also provide products and services supporting genome editing, synthetic biology, and next generation sequencing. NEB also provides free access to research tools such as REase. What are the types of restriction enzymes? So far they recognized 3 to 5 categories of restriction enzymes. Type 1 restriction enzymes were the first to be identified and were first identified in 2 different strains of E. coli. These enzymes recognize specific DNA sequences but make their cut at seemingly random sites that can be as far as 1000 base pairs away from the recognition site. They have 3 different subunits, HSTM, HSTR and HSTS. They also need the cofactors like ATP, magnesium ions and S-adenosyl methionine. The best examples for the type 1 restriction enzymes are EcoK1, EcoAI, etc. Next, the type 2 restriction enzymes. They mainly protect bacteria and archaea against invading foreign DNA. Type II enzymes differ from type I in several ways, which recognizes and cuts directly within the recognition site. They form homodimers with recognition sites usually undivided and palindromic and 4 to 8 nucleotides in length. They are usually homodimeric or homodetrameric enzymes. As I said earlier, they recognize and cleave DNA at the same site and do not use ATP or aromat for their activity. They usually require only magnesium ions as a cofactor. These enzymes cleave the phosphodiester bond of double helix DNA. It can either cleave at the center of both strands to yield a blunt end or at a staggered position, leaving overhangs called sticky ends. Type II restriction enzymes are the most commonly available and used restriction enzymes in recombinant DNA technology. The most common type II enzymes are those like EcoR1, HIN3 and NOT1. Type III recognizes specific sequences but makes their cut at a different specific location, usually within about 25 base pairs of the recognition site. Type III enzymes, they have two subunits, MOD responsible for DNA recognition and modification, RES responsible for DNA cleavage. They recognize two separate non-palindromic sequences that are inversely oriented. They cut DNA about 20 to 30 base pairs after the recognition site. These enzymes contain more than one subunit and require aromat and ATP cofactors for their roles in DNA methylation and restriction digestion respectively. Some of the examples are EcoP1I and EcoP15I. Next come is the type IV restriction enzymes which recognize and cleave methylated DNA and they are not part of a restriction modification system. Type V restriction enzymes, the best example are the Cas9 guide RNA complex from CRISPR. They utilize guide RNAs to target specific non-palindromic sequences found on invading organisms. They can cut DNA of variable length, providing a suitable guide RNA. The flexibility and ease of use of these enzymes make them promising for future genetic engineering applications. Then the artificial restriction enzymes, they can be generated by fusing a natural or engineered DNA binding domain to a nucleus domain. This is an interesting table which represents the most popular restriction enzymes with their specific cutting sites. Most of the recognition sequences are palindromic. The cleavage patterns are also described for HIN3, SMA1, EcoR1 and BamHI. Here you go. These are the steps involved in DNA binding and cleavage by a type II restriction endonuclease. That means by BamHI. This specifically shows the non-specific binding and how the specific binding looks like. Then the coupling followed by catalysis and product release. Can we make a restriction mapping of a DNA fragment with the help of the restriction enzymes? Yeah, exactly, we can able to make it. This is a simple example. Here we use restriction digestion with a single enzyme, which means two different enzymes individually and double digestion with the two enzymes. If we map the digested fragments, it shows the restriction analysis of the DNA fragment. KPN1 alone produced a large fragment and 1,000 base pair. BamHI gave a large fragment and 2200 base pair and 600 base pair products. Therefore, if we digest with both these enzymes, we may get a large fragment, a 1000 base pair fragment, 1200 base pair fragment and a 600 base pair fragment, and we can make up a restriction map like this. DNA methylation is an important means for studying restriction enzymes and their functions. Bacteria use restriction endonucleases to degrade foreign DNA introduced by infectious agents such as bacteriophages. To prevent the destruction of their DNA by the restriction enzymes, bacteria mark their own DNA by adding methyl groups is called a methylation. They catalyze the transfer of methyl groups from S-adenosyl methionine to specific nucleotides of double-stranded DNA molecules. Methylases are often used in molecular cloning to protect DNA from digestion during DNA cloning or cDNA or genomic library construction. The next DAM and DCM methylases. DAM methylation regulates post-replication mismatch repairs, transfers a methyl group from SAM to the N6 position of the adenine residue. DCM methylase, also known as MEC, transfers a methyl group from SAM to the internal cytosine residue. Thanks for watching, and these are the references to read. Thank you so much.